All right, welcome to the mysteriousness of the chylomicrons chapter. This is introduced in chapter 24, but is talked more about in detail in chapter 25. You've listened to other lectures on chapter 25 by Dr. Jen, but I'm going to spend some time highlighting this figure in particular, which probably means it's important, which probably means you should take some good time to really understand what's going on in this figure. And so we're going to talk about lipoproteins, which are the carriers of lipids, and the organs associated with carrying these lipids. So we start with number 1, and I'm going to walk through the image in order. So we start with number 1 with micelles. We talked about micelles in chapter 24. These are a single bilayer membrane filled with a little bit of triglycerides, which are the monomers of, or fatty acids, and triglycerides, which are the monomers of lipids. They go through the small intestine epithelium. They get absorbed there, and then they get repackaged as chylomicrons. And the difference between micelles and chylomicrons is micelles have really boring membranous structure, which allows them to easily diffuse into the cells of the small intestines. But chylomicrons are special because they have little proteins on them that help flag them and say, like, this is what I have inside me, this is what I have inside me, and so on. And they also have some vitamins covering their cell surface as well. So once they get exocytosed as chylomicrons from the small intestine, then they are absorbed into the lacteal. They do not go into the blood vessel, into either blood vessel, because they are too big and they're just too big. They won't fit in the blood vessels, but they do go into the lacteals, and the lacteals are part of the lymphatic system, and they're not too big for the lymphatic supply because they can actually squeegee their way in. So now the chylomicrons are traveling around through the lymphatic system, and if I can remind you, the lymphatic system ends all. Here, let's draw a little terrible person here. Oh, all right, uneven legs, perfect. So the lymphatic system, everything from this part of the person drains into the thoracic duct. And then everything that drains on the right side drains into the right lymphatic duct. But both of these can dump into their respective right subclavian right subclavian vein, and then the thoracic duct dumps into the left subclavian vein. All right. So now that we're in the subclavian veins, now we travel through the vasculature. Okay. Once we get into the capillary, this is really neat. The chylomicrons will actually physically bump into the walls of the capillary, and when they bump into the walls of the capillary, those specialized proteins on the cell surface of the chylomicron tell the blood vessel, hey, I'm a chylomicron, and the blood vessel responds by releasing lipoprotein lipase. So lipase because we're talking about fats, ACE because it's an enzyme and has to break down those fats. Lipoprotein, lipo referring to fats, and protein referring to the protein parts of the chylomicron. So this is a really neat feature. And this is why it's really important to have these packaged as chylomicrons first before they travel to the bloodstream. If it was just a micelle and it bumped against the blood vessel, it wouldn't do anything because the micelle is just made up of lipids around the cell surface, and it wouldn't even register. Like, the endothelium of the blood vessel would just think, like, oh, okay, whatever, there's just some phospholipids bumping into me, nothing special. So whenever that happens, then the other thing, once the endothelium releases lipoprotein lipase, that'll be really important because at this point, the chylomicron, may I remind you from chapter 24, got flagged with an enzyme called lipase. Let's see here. I think, lingual lipase, and there's pancreatic lipase. So whichever lipase it is, the lipase comes in, actually flags the molecule. And then this flag is really important because lipoprotein lipase, which is a different type of lipase, needs to come and actually read that flag, and only then can it actually help break apart the insides. So that's why we mentioned lipase earlier. Okay, anyway, so now we have all of the enzymes that we need to break apart our chylomicron. So in the chylomicron, we have triglycerides and fatty acids. So the fatty acids go to the adipocytes, and any triglycerides that we need from the fatty acids we can make. Whatever triglycerides we already have can actually be burned by the skeletal muscle to make ATP. So this is going through the glycolysis reaction, or cell process, I shouldn't say reaction. And so we make ATP so we can keep moving. Awesome. All right. So what about producing more chylomicrons and storing more triglycerides for later? Like, what if we don't need to use them immediately? So that's why we go down into the liver. So remember, we're traveling through the vasculature, so we can either enter here through the hepatic portal system, or maybe we entered here through proper hepatic artery. We get into the liver. In the liver, we're going to do one thing first. We're going to break apart the chylomicron, take out the triglycerides, use those triglycerides so that the liver can keep doing what it's going to do. Then it's going to package another chylomicron, add in some cholesterol, and shoot it out through another lipoprotein called a VLDL, very low density lipoprotein. And those are going to continue to circulate around the blood vasculature and help keep supplying adipocytes and skeletal muscle tissue as needed. That's part one. Part two, the chylomicrons, we'll break them down, we'll use those triglycerides, we'll repackage them with a little bit of cholesterol, and instead these are going to be low density lipoproteins, LDLs, and those travel through the bloodstream and actually go and target peripheral cells that are not adipose and muscle cells. And the lysosomes in there are going to break down the walls of the the lipoprotein, and take out the cholesterol and use it for different various reasons. We could use it for building of certain steroids. Oh goodness. So as this chapter teaches you, cholesterol is a precursor for a lot of really important steroids like testosterone, progesterone, estrogen, etc. We can also use cholesterol to help maintain stability of any lipid structure. So like this, the plasma membrane. And various other things, but those are the two main ones that we really care about. So cholesterol will either be used there in the cell, and whatever cholesterol is not used goes back into the bloodstream and then gets picked up by a continuously circulating lipoprotein called an HDL, or a high-density lipoprotein. HDLs will carry those cholesterol molecules, bring them back to the liver to be recycled and used again in the LDLs and the VLDLs. And then whatever is left of the HDL will continue back into circulation, waiting to pick up more cholesterol, and so on and so forth. Okay. So HDLs, LDLs, the one is bad, the one is good. There's no such thing as good. So the LDLs are usually the ones that are considered the bad cholesterol, and the HDLs are usually the ones that are considered good cholesterol, but that's kind of a misnomer. Neither one in high quantity is really good for you, but people tend to, or I shouldn't say people, but originally whenever we were first understanding what cholesterol does and how it works in the body, high levels of LDL tended to indicate that those patients would end up with they had some sort of cardiovascular risk or some sort of prevalent heart disease at the time, because, and this makes sense, because there was so much cholesterol that those LDLs had to continuously shuttle it around. So we just assumed that high LDLs, they probably had a lot of cholesterol in their system. It was only just a matter of time until something went wrong. A plaque built up, a vessel got clogged, etc., etc. The HDLs are better at holding on to cholesterol and then letting it go, versus the LDLs actually have to enter a cell, and it's a more complicated process. So that's why the HDLs were considered the good cholesterol, but they also have cholesterol. So having high levels of HDL doesn't necessarily mean that you're in good health. It also means that you have more HDLs because you probably have more cholesterol circulating, so you need more HDLs to continuously circulate all that cholesterol. Our body cannot cannot, I repeat, cannot break down cholesterol at all. There's no mechanism for us to do that. Once it's cholesterol, it's cholesterol. We can convert it into other things, but we can't ever break apart that molecule and use its carbon components. So once it's in there, it's in there. We have many mechanisms for, I shouldn't say many, we have one really well-studied mechanism for making a lot of cholesterol, and this is a continuous mechanism that's happening all of the time. And of course, you ingest a lot of these fats as well too. So when you have these transport lipoproteins in high numbers, that also probably means you have a lot of cholesterol that needs to be circulating through your bloodstream. So it's not appropriate to think of LDLs as being good cholesterol and LDLs being bad cholesterol and HDLs being good cholesterol. Please don't continue that logic. Be familiar with what the two do. But that's it for this slide. Be familiar with this story and be able to tie this into how we actually digest lipids, which comes from chapter 24.